I'm going to introduce Dean Williams. Dean's going to carry the conversation forward. Dr. Dean Williams got his education in ecology from Purdue University. He's primarily studied population biology and behavioral ecology. A lot of this information helps inform management of our invasive species, and he'll talk specifically about a molecular survey of the recently introduced genotype of hydrilla in the Connecticut River. Thank you, Dean.
So, thank you for the introduction. I'm going to talk today about molecular survey for a recently introduced genotype of hydrilla in the United States, as well as some work with the monoecious biotype. And, you know, this map has been shown before, but we know about the dioecious and the monoecious biotypes here in the US. The monoecious genotype, it's native to Eastern China, South Korea. It's most likely where it came from. The dioecious genotype occurs over a fairly large area from India, Pakistan through China, and also in maybe some other places as well. Worldwide, hydrilla can be grouped into 4 main genetic groupings, call them clades, A, B, C, and D. And both the monoecious and dioecious varieties that were introduced into the US both belong to clade B. So the reason for this study was basically this paper here, in which Tippery et al. Discovered that the hydrilla that was in the Connecticut River was something different. And the morphology had seemed different, which is why the genetics were carried out. And when you look at the genetics, we see that hydrilla in the Connecticut River is from clade C, so a completely different genetic grouping than either the monoecious or dioecious biotypes. And so Nathan Harms and Grodowitz and I had done a previous study in which we were doing a genetic survey across Eastern China and South Korea to look for monoecious populations there in the native range to help with looking for biocontrol agents. And so we had this data set of genotyped hydrilla across Eastern China as well as in South Korea. And so we wanted to compare this new introduction into the Connecticut River with our data set. And so over here on the right, or left, sorry, what you see are 2 haplotype networks. One is for clade B and one is for clade C and D. Each one of those circles is a different genetic type, and this is based on the chloroplast DNA. And the green circles inside of the white circles, those indicate the ones that have been introduced into the US. So the monoecious variety is down there in clade B, mono 2121, and dioecious is 226 up there at the top. And we were using an additional marker to Tippery that gives us a little bit, better geographic resolution in terms of these different genetic types. And so when we do that, the exact match that the one from the Connecticut River came out to be was 17-8 there in the clade C network. So you'll notice those circles, the size of the circles just indicates how common that genotype was in the native range. And then the lines connecting are the different number of genetic mutations that separate those different genetic types. Okay. So if you look over at the map at the right, what we did was we then mapped out where 17-8 was found, as well as clade C in general. And we see that 17-8, it's these open triangles here, and they occur up there in northern China. So it's definitely a temperate adapted plant and can withstand cold temperatures. It's also closely related to some other genetic types that have been found in far northern areas as well, in Japan and some in northern Europe. Northern Europe and Siberia and those areas have been very poorly sampled. So, we've only found this direct genetic match from northern China, but it doesn't mean that it didn't necessarily come from somewhere else, because a lot of those northern areas haven't been sampled as well. So clade C in general, not just 17.8, but clade C overall occurs throughout China. That's all these triangles, both the closed and the open that you see. Dioecious, just like in the US, tends to have a more southern distribution in China. And the monoecious is kind of here in the middle. It occurs very patchily in the native range. It's not actually a very common genetic type in the native range. Okay, and we have found some evidence in China that clade C can hybridize with clade B. Whether that would happen here in the US, we don't know. But that might be another possibility for bad things to happen potentially, I guess. So the main point of this project has been to determine the geographic extent of this new introduction by sampling the Connecticut River, as well as other water bodies within 50 miles of the river, and to determine the genetic diversity of that new introduction, as well as kind of complete a survey of the different biotypes that occur in water bodies in the Northeast, and water bodies that are of concern for the U.S. Army Corps. And that's who's funding this particular project, actually. So this last year, and this is going to continue on into next year, but last year we received 361 samples. This is a breakdown by state, as well as the people that collected them or helped with the collections. And so the process involved, extracting DNA from that collected hydrilla, and then genotyping them at a quick chloroplast marker that we made that can distinguish clade C versus mono versus dio biotypes. And we also genotyped all these samples at 7 nuclear microsatellite loci, which are much more variable genetic areas that allows us to then determine if there are different clones of these hydrilla biotypes. 348 samples were successfully genotyped. So there's about 13 that the DNA quality just wasn't good enough for whatever reason. Okay. So for the chloroplast results, what you're seeing here is a map of all the places that were sampled, and all of the green and M points on here, those are all the monoecious biotype. And the C and the yellow points, that's all clade C hydrilla, and you can see how it's concentrated there in the Connecticut River. Okay, so there was 330 samples of monoecious, and 18 samples were genetically characterized as clade C. Okay. So only clade C has been found so far in the Connecticut River. The last talk I gave on this, there was a couple samples in here that looked like they were monoecious. I went back and looked at some of these more carefully. There was some samples from the Connecticut River that the DNA quality wasn't very good. I went through and re-extracted all these to double check, and the couple that had come out as monoecious actually had originally had very low DNA quality. Most likely that was due to some low level of contamination, I'm guessing. But definitely it would pay to do more sampling along the river and the tributaries to see if it really is all clade C in there. But so far, it looks like it's all clade C with the samples, the 18 samples that we have that worked. So if we look at the nuclear microsatellite genotypes, all clade C individuals, all 18, had the exact same genotype, which suggests that it really is a single clone that was introduced. So not a lot of genetic diversity there, just a single genotype. Monoecious, on the other hand, we found 57 genotypes, but most of those belong to a single clone. So that's what you see in the bar graph here. Down on the x-axis, that's just clone ID number, and then the bars represent how many individual samples belong to each of those clones or each of those different genotypes. And the first one there was by far and away the most common. 250 samples actually belong to clone 1. Whereas you can see there's a few others in which there were multiple individuals, but the vast majority are just due to a single individual having that genotype. Okay. Now, most of those are very closely related, so this suggests that really there probably only was one original clone that was introduced, but it has since diversified. So what you're seeing here is this genetic distance threshold on the x-axis. We start out at a genetic distance of 0. In other words, we're not taking into account that any differences might actually be the same. There's 57 clones there. And we can calculate kind of a measure of haplotype diversity, which that number, that H of 0.43, all that means is if you were to go into this area where we sampled and you pulled out randomly 2 different hydrilla plants, there would be a 43% chance that they would have different genotypes, right? Now, if you compare that to what we see in the native range, it's actually about a 90% chance. So, not surprisingly, the introduced population has lower genetic diversity. And even if we take into consideration, let's say, okay, all of those single off genetic differences, are due, they really derived from the same clone, you then drop down to there being 6 different clones, and H goes to 0.04, so really low diversity. But if you do that in the native range, you still use that genetic threshold, it's still around 0.9. So a lot more diversity occurs in the native range there. So the question is, why in the world do you have all of these different genetic types that are, in a genetic sense, they're very similar to each other? Well, one thing that can generate that when you genotype clonal plants, it could be genotyping error. There's always error in everything that you do, and the question is that error high enough to change your results? I have regenotyped some of these, and they do come out with the same genotype. So, and some of the previous estimates of error that we've calculated for our genotyping is fairly low. So, some of those clones, some of those 57 clones might be due to error. But more likely, most of them are probably due to what's called somatic mutation. So, this is probably what has led to fluoridone resistance in the dioecious type down in Florida. It was probably due to somatic mutation. A mutation occurred in the gene that that herbicide targets and made them resistant to it. But in hydrilla, basically, because it has high growth and can grow very quickly, all that means is, as DNA is replicating and the plant's growing, there's more opportunities for mutation to occur. And this is not an uncommon thing to happen in plants. A classic thing in the past that people had noticed is if you genotype one side of an oak tree and then you genotype the other side, you'll often find small genetic differences, even though it's the same tree. That's because plants have that modular growth pattern. And so you can potentially get different mutations in different parts of the same plant. Now, for something like hydrilla, that could be important, say, for instance, when there was somatic mutation that led to fluoridone resistance, those parts of the plant that had that mutation can fragment off and then start new plants. And so you could potentially get the spread of favorable mutations, with this kind of a situation. Whether that would happen in the monoecious one or not, of course, we don't know. One interesting pattern that I noticed looking at these different genetic types is oftentimes they're clustered in certain areas. And this is just a few examples here. That up here on the left top, that's Creamery Pond. The green markers there, that's the most common type, the 250 individuals that were in clone 1. But all of those little white pins that I put down, each one of those is actually a different clone. And so Creamery Pond had pretty high diversity for those clones. Now remember, those are not very far apart genetically from each other. Over here at Nakamixon, you'll see a bunch of pink ones. So that pink clone only occurred in Nakamixon, as well as some other types, and there was none of the most common type in that area. Down here in New Croton Reservoir, it had mainly the common type, although there's a few others in there as well. And similar kind of thing over here in Pinchot Lake as well. So I'm not sure what that means. It is kind of interesting that some of these things are clustered like this. Could certain areas be resulting in higher mutation rates? I have no idea. This is something that I think will be interesting to look at in more detail and as we get more samples in the future. Overall, if you look at kind of the pattern geographically, individual samples that are located close together are more similar genetically than those that are located further apart. And this is just a correlogram that's illustrating that. You have distance classes on the x-axis and genetic correlation of individuals in each distance class. And so it's all pairwise comparisons of, for instance, individuals that occurred within 1 kilometer of each other or 5 kilometers of each other. And you can see that at 1 and 5 kilometers, they're above the red dotted lines, which are the 95% confidence limits around the random expectation in this case. So there's significantly more similar to each other than they do once you get further away. Also broke this up just out of curiosity to see across states what the genetic diversity was like in the monoecious and using all of those different clones that we found. And this would probably be better to do by watershed actually in the future, but here you can see in Connecticut, and this is not including the Clade C, this is just including the monoecious. In Connecticut, there was an H of 0, meaning they were all identical. Massachusetts had a little bit more diversity, and then Pennsylvania actually had the most genetic diversity, which is kind of interesting. I'm not sure, again, what's leading to these patterns, and it's something I'd like to look into more in the future. Don't know if this kind of variability could be important from a management standpoint. The markers that we're using to generate this variability or to look at this variability are not, directly linked to things like herbicide resistance or anything like that. These are neutral markers. They're good for potentially following individuals and how individuals have been moved around and things like that. But they don't necessarily say anything directly about things like herbicide resistance or anything. But if mutations have ever resulted in that, these could potentially be linked, to those genetic changes, and they could be used as a marker potentially. So it may be that in the future, it might be useful to think of these things at the clone level if it looks like that it's important. Okay, so for this year, really would like to get a lot more samples from the Connecticut River and surrounding areas. Especially, it sounds, as Greg talked about all the tributaries going into the river, it would be interesting to sample those as well as other areas where the monoecious biotype occurs in the Northeast, and talking about in North Carolina and places like that as well. Okay. And that's all I have for today. Does anybody have any questions?
Thanks, Dean. I'm sorry I failed to mention that you're with the Army Corps in the ERDIC
That's okay.
department in the Engineer Research and Development Center, which is a pretty
Yeah.
critical piece to mention. So I'm glad that we have the logo here. And I do need to just mention that the Army Corps has been extremely involved and responsive to requests from the Northeast Aquatic Nuisance Species Panel and members of our society for looking into this Connecticut River specific genotype. And I want to thank a lot of the folks from the different states from the NEANS panel region for helping to coordinate and obtain and send in the samples for analysis. So this is definitely a huge partnership effort to look at this rather complicated story. And I really appreciate the light that you're shedding on this, Dean. It's really fascinating. We do have a few questions. The first one comes from Jack Andrews. He's asking, have you considered looking into polymorphisms performing such as RAPD to see if a difference genetically can be determined with the monoecious genotype?
Like using rapids?
I think
it's what, yeah.
Yep.
Yeah, we used microsatellite loci instead, just because we had those developed for hydrilla. They may not be quite, I guess what I would maybe want to, try next would be AFLPs, which are kind of like rapids, but they're easier to reproduce the profiles. So, for the microsats, we sampled 7 different genetic areas. If you do something like AFLPs, it's probably sampling like 100 different genetic areas at once. It's kind of a shotgun approach. It's good for looking at clonal lineages. I mean, it would be interesting, I guess, to look at some of this data to see if there's even more clonal lineages if you look at those AFLPs. But
we will be looking at gathering more samples, of course. And
okay.
I know there was a previous question about have we looked at, yes, AFLP, a great technique. Thank you. Okay. Yeah, we will be, hopefully the Army Corps will get involved in doing some mesocosm studies in the near future so that we can get going on some kind of containment and treatment.